Hello, my name is Xandre Clementsmith, and I will be presenting dynamic network activation of hypothalamic neurons in REM sleep. So we began with data from the Medical University of South Carolina. They conducted a study that tried to correlate the activity of a network of melanin-concentrating hormones in the hypothalamus with various activities of a rat. In particular, they found that REM sleep and exploration showed activation in the hypothalamic network. We decided to take this a step forward by taking the original data and trying to reconstruct the actual network rather than just finding a correlation. So looking at the individual neurons and how they connect to each other. You can see an example of what this network might look like in figures 1D and 1F. It shows that there's a group of neurons that are showing a calcium fluorescence, basically a correlate to their actual activity, and gives a hint of how they might be connected. So we start with the individual calcium fluorescence of each neuron. As I said, this is a correlate. It doesn't show exactly when the active potentials are, just hints when they might be. So as a result, we use a MATLAB package called MLSpike to go ahead and extract these action potentials, or the spike train, from the continuous calcium signal. This allows us to use the data for many more methods, as well as for nonlinear methods that deal with not just simple causal connections. For instance, if two neurons are being affected by a third neuron, it might be able to find out whether these original two neurons are affecting each other or just being affected by a third source. So we managed to get all of our data processed through the MLSpike algorithm and collected a discrete action potential train for each of them. We then decided to use a general linear model approach to go ahead and reconstruct the network. So we would take a majority of the neurons and use them to predict a final neuron just given a series of weights, basically the amount of connection between individual neurons or how much they are probably affecting each other. In figure 3A, you can see a scatter plot of the actual and predicted values. So you can see it generally follows the line of identity, where a predicted value usually matches an actual value with a little bit of noise. Unfortunately, as it gets into the negative numbers, it skews a bit, which I can show you here on figure 3B. There is a jump in the Y prediction below the Y actual. Why this occurs may have to do with the algorithm. It may have to do with the network adjusting in an odd way that our algorithm doesn't account for, because it is just a linear algorithm. In addition, you see that there is a jump or an inversion at the end of the prediction and actual, where one will go up and the other will go down with a bit of a delay. Again, this might be an error in the model, but it's much more likely that it's a characteristic of the network, something that will need further investigation. So we found a lot of correlation between the point process, the action potentials, and the continuous signal. In the signal, in the figure 2, the signal had a correlation coefficient of around 0.67, so a pretty high correlation between the action potentials and the spikes. You can see that fairly easily visually here, showing that there is quite a lot of overlap between the two, especially for a continuous and a discrete sample. The general linear model we found didn't perform super well with the action potentials. This is probably due to the sparsity of the data, though that sparse data will be very useful for further methods that employ more action potentials. It also didn't work as well for the Poisson distribution, and the normal distribution was the best one to actually get a prediction. However, this isn't necessarily the case that it's the best way to get the weights, just one of the best predictors of the weights. Finally, we tried several other methods that are currently in further process, but one of the major ones was transfer entropy, which allows us to take the information from one neuron to another. We're also trying several nonlinear approaches to continue this research into the future. I'd like to thank the National Institute of General Medical Sciences and the National Institute of Health, the South Carolina Embrace Scholarship, and thank you very much for listening.